Hi, and welcome to the MIT Faculty Forum. I'm Karen Weintraub, a Cambridge-based freelance journalist, and I will serve as the moderator today. As a reminder, we welcome your questions during the chat. For MIT alumni joining us via Zoom, please use the Q&A feature found at the bottom of your screen. For all others listening on YouTube, you may add your questions to the comments field. We also encourage you to tweet using hashtag #MITBetterWorld. We will get to as many questions as we can. Today, we'll be hearing from two experts who will share some of their current work on ingestible sensors and smart pills. Let's start with an overview from each panelist. First, Dr. Gio Traverso. Dr. Traverso is an instructor of medicine at Harvard Medical School and a gastroenterologist in the Division of Gastroenterology at Brigham and Women's Hospital. He completed his PhD thesis on novel technologies for the non-invasive detection of colorectal cancer in the lab of Bert Vogelstein at Johns Hopkins University. For his postdoctoral research, Dr. Traverso transitioned to the fields of chemical and biomedical engineering in Professor Rob Langer's lab at MIT, where he developed a series of novel technologies for drug delivery, as well as physiological sensing via the gastrointestinal tract. His current research is focused on developing systems to enable efficient delivery of therapeutics through the gastrointestinal tract. He continues to collaborate closely with Professor Langer and is a research affiliate of the Langer Lab at MIT. Dr. Traverso?
Thank you so much, Karen. I'm just going to share my screen with the audience. I hope everybody can see this. And what I wanted to do was just give you an overview over the next few minutes and then allow for questions. And let me just start off with this, this photograph of C. Everett Koop. And I think that one of the key messages from C. Everett Koop really encapsulates one of the most significant challenges that we face as physicians, today, and not only here in Boston, but really globally. And really, I think he said it, perfectly, and that is the drugs just don't work if people don't take them. And that challenge, that problem of non-compliance or non-adherence, is something that's been written about broadly in multiple outlets, including, for example, the New York Times. And as a community, as a society, we're just not very good at taking medication. And actually, non-adherence manifests in excess of 100,000 deaths per year in the U.S. alone. And in the U.S., the American healthcare system is responsible for expenditures in excess of $100 billion annually. Moreover, during my time researching and developing technologies to help address this challenge, one of the most compelling quotes that I've come across is the one that I include here at the bottom, and that is that increasing the capacity or increasing adherence rates really stands to have a much greater impact to society than the introduction of new drugs in several disease areas. And so with that in mind, we really set out to do what you see here, and this is a cartoon from The Economist that really helps encapsulate what our group really is trying to do, and that is to create a metered-like system that has the capacity to dose medication over a prolonged period of time that is orally delivered, hopefully in a smaller form factor than the one demonstrated here. And so we actually did this, and we developed a system that looks much like a star that can fit in a capsule. And this opens up in the stomach and can subsequently be retained for a prolonged period of time. By that I mean anywhere from a few days to multiple weeks, and all the while releasing drug from the dosage form, and then subsequently break up and pass through the gastrointestinal tract and be excreted. And so, having developed the ability to safely reside in the gastrointestinal tract, we loaded these systems with a drug. The first drug actually that we evaluated is a drug called ivermectin. And ivermectin has been used broadly in the field for treatment of a range of parasitic infections, but more recently has been observed to be toxic to mosquitoes. And in collaboration with the Gates Foundation, we developed a system that could release ivermectin over the course of a couple of weeks to really provide or amplify mass drug administration campaigns against malaria. And we published this work at the end of last year in Science Translational Medicine. So having achieved this, and in parallel, we were thinking about different systems that we might be able to consider to really help diagnosis and really arrive at treatment and optimal treatments for a broad range of conditions. And we started by really thinking about how we look at the fundamental vital signs today. And in this slide, what this really captures is what most folks would encounter either in the primary care physician's office or in the hospital with respect to how vital signs are typically evaluated. And that's typically using some kind of transdermal or even visual evaluation. And so what we wondered was, could we create an ingestible stethoscope, and essentially could we capture an acoustic wave form from within the GI tract, and from that, specifically tease out the heart rate and respiratory rate. And in collaboration with the team at Lincoln Labs, we actually did this. We captured the acoustic wave from different portions of the GI tract, and this was initially done in a pig model, and then extracted heart rate and respiratory rate. So we showed that we had the capacity to sense at least these fundamental vital signs. And so, as we were developing these other systems and demonstrating the ability to reside in the gastric cavity for a prolonged period of time, we started wondering about the potential for prolonged monitoring using an ingestible capsule. And, as one starts to wonder and sort of consider this possibility, one of the challenges that we face when doing this is how do we power such systems? So if we had a system, for example, reside in one's stomach for, let's say, weeks or even months, a battery may be insufficient to really provide the needs of such a system. And so in collaboration with Dr. Nadeau, who's the other speaker today, we took a page from our old high school or school textbooks and developed essentially a battery that's really inspired by the lemon battery, but instead of using the acidic lemon juice as such as the electrolyte, we use the gastric fluid as the electrolyte. And using that system, we developed prototypes and demonstrated that we had the ability to sense and measure a specific vital sign, in this case temperature, and communicate that to several meters away over the course of a week. I know Dr. Nadeau will be diving into more detail around these systems that we developed together. Now, we've also looked at other systems to try and power electronics from that may reside in the gastric cavity or other portions of the gastrointestinal tract. And one of the other areas that we've looked at is midfield powering systems. And so here we actually constructed some new antennas that had the capacity to be powered externally. And so, with that, and I just want to leave the conversation with this last slide as some food for thought, as we at least consider this area and the tremendous potential that we see in positively impacting and really changing how we engage with patients and how we keep patients and folks really out of the hospital. And so, I've broken up in this slide into, I think, two key areas. One is drug delivery. And so some of the gastric resident forms that I've described really enable ultra long dosing. So that's potentially having systems that reside in the body for many weeks or potentially someday even months following a single administration event. And those may be used for either local or topical therapy. The incorporation of electronics may enable triggerable therapy, and perhaps one day systems that have closed loop systems that are linked to a specific sensor and specific either analyte or vital sign to essentially couple the actual drug delivery event. And then, of course, with respect to sensing, I've mentioned sensing of vital signs, but there's a tremendous breadth of opportunity here with respect to small molecules and macromolecules. And so, for example, analytes or toxins that we may want to sense, and again, potentially someday couple to an actual treatment intervention. And some of the challenges as we sort of contemplate and consider these different options really have to do around communication. How do we maintain those communication channels encrypted, for example, but then also how do we maximize those in different settings? Integrating all of these systems together, and particularly in a form factor that is compatible with gastrointestinal transit. And then as far as implementation, how do we integrate these systems into current healthcare models? And so are these systems that we would use to maintain and help keep folks healthy at home, and how would those interface with the current standard and sort of office practices? And then with the ability to monitor folks for longer periods of time, there are certain challenges from the big data perspective that start to arise and really present very unique opportunities on health sort of population diagnosis and understanding, in a broad set of settings. And so with that, I just want to acknowledge the tremendous the tremendous group and efforts. And this is our group at our last year's summer retreat. I'll leave with that. And I know we're going to transition to Dr. Nadeau, but I'm happy to take questions after, towards the end. But in case anyone has any questions, these are my emails. By all means, feel free to get in touch. Thank you.
Thanks so much. That's really amazing work. Just a reminder, the Q&A button, I think I described it wrong. I think it's at the top left of your view. And so please put your questions there. We will address them after the next speaker, who is Dr. Phillip Nadeau, a postdoctoral researcher at MIT, though he's heading off shortly to join Analog Devices as a research scientist. Congratulations. Dr. Nadeau works at the intersection of ultra-low energy electronics and biomedical applications. He completed a master's and a PhD in electrical engineering and computer science with Professor Anantha Chandrakasan at MIT, the lab in which he still works. And he holds a bachelor's degree in electrical engineering from the University of Waterloo in Canada. He also completed internships at Texas Instruments and Intel. He's going to talk more about that project that Dr. Traverso just mentioned.
Hi, Karen. Thanks so much for the introduction. Very excited to be here. Let me just see if I can get the slide shared here. Okay, here we go. So as Karen mentioned, and as Dr. Traverso alluded to, I'll present just a quick overview of work that I did along with Professor Chandrakasan and many collaborators in collaboration with Giovanni Traverso's group on looking at prolonged energy harvesting for gastric devices. And something that we came up with in collaboration between my advisor, Professor Chandrakasan, and Professor Traverso, or Dr. Traverso. And this is in the area of ingestible electronics. And we've seen a number of commercial examples of ingestible electronics over the last few years. Perhaps one of the most recognizable is the pill cam, which takes pictures of the GI tract and transmits those pictures wirelessly. More recently, Philips Healthcare, which is now Medimetrics, has been developing an ingestible capsule for multimodality sensing, pH sensing, and also temperature sensing, as well as drug delivery. What we can't see in these pictures, but what is clearly contained inside is one, or in some cases two, relatively large button cell batteries, which are used to power these electronics. And for reasons of toxicity and also for safety and cost, what we decided to look at was, could we power ingestible electronics with energy harvesting? So in other words, pulling chemical energy out of the GI tract, the stomach and the intestine, and using that to power relatively sophisticated electronics. So we want to know, can we make electronics low power enough to be powered off of such a cell? And also, how much power would that cell generate? What are the statistics of it? How long it would last? Questions of that nature. And looking at the literature, we see a couple of examples of in vitro experimentation on biodegradable cells using synthetic gastric fluid. There's also a commercial example, one of the first, if not the first, in vivo demonstration of this kind of cell by Proteus Biomedical, where they had a self-powered ingestion monitor that would wake up upon ingestion, harvest a small amount of power, and send a packet to indicate that the person had taken their medication to a body-worn patch. And they harvested for a couple of minutes. Motivated by the kind of work that Dr. Traverso has been doing with long-term drug delivery, we wanted to know whether we could use this kind of technology for long-term monitoring from inside the GI tract. And so the first thing we did was set out to characterize a biogalvanic cell in the GI tract by making a custom capsule out of commercial electronic parts. The first generation capsule was a battery-powered capsule. We can see it has a microprocessor inside and also a potentiometer that we could use to characterize the cell. And in this case, we chose copper and zinc electrodes as the cell. The zinc particularly for its longevity as compared to the magnesium that some of the previous work had used. And so with this first electronic hardware, we were able to perform experiments in a large animal model in pigs by depositing this capsule inside and having it transmit wireless packets with measurements of the voltage and power obtained from the cell. And so the capsules were put endoscopically inside the stomach. You can see a picture of a capsule here in gastric fluid. And the capsules transmitted wireless packets at the 900 megahertz frequency to a receiver that we had connected to a PC for logging. And the transmission distance was a couple of meters. And so the animals were able to go about their normal routines, feeding, eating, sleeping, and walking around. So it's truly an ambulatory measurement in that sense. And here's an example of what we're able to characterize from this system. And so the top plot shows the voltage obtained from the cell on the order of 150 millivolts or so. And then the lower plot shows an example of the peak power obtained from the cell. And these measurements were performed over multiple days. The capsules were retained inside for up to about 7 days on average, and then subsequently passed to the small intestine. We can see an example here confirmed via X-ray of passage from the stomach into the intestine, and showing a drop in power in the intestine subsequently. More on that in a second. Now, if we look at the statistics of the power that are available, again, on the order of about 150 millivolts or so, we see a decline in the intestine. The lower plot is showing the statistics of the power on the order of about a microwatt per millimeter square of electrode area. We see a drop in the power when the capsule moves to the intestine, but we're still able to measure it, which is one of the first demonstrations of power from a biogalvanic cell in the intestine, on the order of 10 nanowatt per millimeter square or so, and much lower, of course, due to the constricted nature of the environment and the slower diffusion of products and reactants to and from the electrodes, but still measurable. And so this first step was critical to help us understand how to design a harvesting system that would actually harness this energy and do something useful with it. And so the second part of the work was designing a fully self-powered harvesting capsule without a battery inside. And this capsule is shown here. It looks kind of similar to the other one, except now we've removed the battery and we've replaced it with an energy harvester integrated circuit, which would pull energy from the zinc and copper electrodes and boost it up onto a temporary storage capacitor, which would essentially buffer the energy and allow us to use it periodically to perform a temperature measurement, some computation, and then wireless transmission out the antenna. And this bottom plot here shows an example of the timescales of how this works. This is essentially this is the voltage on the storage capacitor. You can see that over about 10 to 20 seconds or so, the voltage on the storage capacitor is building up. And then during the packet transmission, where the system wakes up, performs a temperature measurement, and then transmits a packet, we can see the drop on the storage capacitor due to all of that energy being used to transmit the packet. But this gives you a sense of how the system works. And then we actually deployed that in an animal. We deployed 3 devices in separate animals. And this top trace is showing an example measurement for one of the devices where we characterized the power density harvested about 150 nanowatt per millimeter square after all conversion losses taken into account. And what we were able to do with that power was transmit the temperature measurement from inside the animal, and that's what you see on the lower trace. You could see some variation during feeding times and drinking times. You can also see certain times for which the power was not available. That's these gaps here. But on average, across 3 experiments in 3 separate animals, we had 200 nanowatts per millimeter square, which allowed us to send packets with the temperature measurements every 12 seconds on average for as long as 6 days. And perhaps temperature measurement is interesting, but is there more we could do? And indeed we can. And one of the further things we demonstrated is that we could do drug delivery using this harvested power. This was done in an in vitro model using real gastric fluid harvested from the animals. And what we see here is an example of a drug delivery device that contains a gold membrane or that has a gold membrane, and inside contains a drug. In this case, we were delivering a blue dye to the fluid so that we could see when it was released. And we could see here in the series of pictures that after putting electrical energy into the electrode and slowly dissolving it away, that over time the electrode develops cracks, and then the drug is released. And I presented kind of an overview, but the full details are available in our Nature Biomedical Engineering paper with the results and statistics for both the power harvesting and for the drug delivery. Where do we go next with this kind of work? We've shown that you could do fairly sophisticated sensing and processing and wireless communication tasks using this kind of harvested energy. I think some of the future work would be improving the quality of the cell. Obviously, we've used a very simple cell. There are a number of examples of cells with higher density that are being worked on now by groups at CMU and also at University of Illinois. And so improving the quality of the cell is one potential direction, and also trying to make the rest of the electronics fully dissolvable. That's an interesting future direction as well. And with that, I'll just quickly thank the many sponsors involved in the various aspects of this work. It was a collaboration between a number of groups, including my advisor, Professor Chandrakasan, Dr. Traverso, and Professor Langer. And with that, I'll end it here, and I understand that we'll be able to take your questions.
Great, thank you so much. And hopefully we haven't cost anybody their appetite talking about gastric fluid and ingestible devices over lunchtime. So Alan Mink asks about the drug delivery. How long does the sensor stay in the GI tract compared to, say, a normal meal? How do we keep it there, and how does it transit out?
So if you're referring to the STAR system, so we've been able to maintain those in animals for multiple weeks, and these are animals that are ambulating and feeding normally. And in fact, one of the key things that we demonstrated in that first paper, and we continue to do a significant amount of work around this, is the ability to really have that dosage form remain intact in across a broad spectrum of diets. And so in the case of the pigs, they're fed a lot of indigestible objects like even banana peels and others, and so we know that from a mechanical perspective, that these dosage forms can withstand a significant amount of challenge and exposure there. And beyond that, I mean, they are eating sort of regular, essentially the chow that's provided, but then lots of fruits and vegetables as well. And then we've done also further testing, looking at, for example, the effects of alcohol and how that might impact on the integrity of the polymer. And specifically, the dosage forms that and the materials that we've chosen are ones that are approved by the FDA and are hydrophobic. So we selected those on purpose, also to minimize fouling of the systems while they reside there. So, going back to the original question, I mean, they can stay there for prolonged periods of time, at least in what we've seen. The longest we've done this for in pigs is 6 weeks, and we remove the dosage form at that point. But in principle, they could be kept even longer. I mean, right now the focus is around 1 to 2 weeks, and then with a view of certain applications around once a month in the future as well.
A follow-up or related question, for Dr. Traverso, can you get the ingestible drug delivery system to have arms, that star shape, that dissolve at a different rate, like one arm each 24 hours? And how many days out can you get?
Yeah, that's a great question. I mean, as you start degrading the macrostructure, then its ability to reside in the gastric cavity starts to change. And so what you can do is essentially have different formulations that are released from each arm. And that's something that we have done, where you could put either different drugs on each arm, or the same drug that is released with different kinetics from each arm to provide either the sustained release or essentially a polypill-like system. But the macrostructure, we want to essentially have the macrostructure dissolve together so that they can be eliminated together, rather than to have a star that has 4 arms or 3 arms, because then we're not able to predict the residence as well.
And how do people swallow the pill, or is it placed in, or when you envision it in people, will it be swallowed?
Yes, it's a great question. I mean, exactly. I mean, it looks just like a capsule. The system fits in a regular capsule. So you actually can't, when you're taking it, it looks like any other capsule you might take. And then it's essentially opens up once it reaches the stomach.
And sorry, how do you control the dosing in this expanded capsule? How often can you dose?
Yeah, no, great question. So that can be tuned, and that really is governed by the indication and also the potency of the drug. I mean, at the end of the day, you have a capsule, and that has a finite volume, and therefore, for a density of 1, let's say, the larger capsules can hold up to 1 gram of material, and therefore, of that, probably between 30 to 50% can be drugs. So that gives you a sense on the kinds of drugs and the potencies that would synergize with this system.
Great. Just a reminder, if you do have a question, please put it in the Q&A. Click on Q&A, maybe in the upper left-hand corner of your screen. If you're on YouTube, in the chat box. Can you give an example of how a feedback loop with an analyte would work? How would this capability benefit patients in the future?
Phil, would you like to take that one? As far as detecting an analyte, for example?
Oh, sure. Let me just make sure it's unmuted here. Yes. So an example might be using chemical means or electrochemical means, or also biological means to detect an analyte in the GI tract. And so we've been working on some engineered bacterial cells that can be used to detect certain chemicals inside the body, and of course you'd need electronics to interact with those, and so if you can do readout from these kinds of chemical or biological analyte detection systems using energy that's harvested from the body, this is obviously a very attractive way to to get in and really sense what's happening in the chemical environment.
Great. What about security? Everybody worries about the security of pacemakers. The questioner alludes to the Homeland TV series here. How much do you worry about the hackability of these ingestible sensors, and what are you doing to address that?
So I could take that one. So security is obviously a major concern, and it's being worked at from a number of angles, some of which is fundamental mathematical approaches to guaranteeing that information isn't revealed, or that the system cannot be taken over. I think the biggest challenge is not necessarily the fundamental approach, but rather the implementation of the system. There are ways that, for example, by measuring the power consumption of a system, if an attacker has access to it, They can go and recover the encryption keys that are used to secure the data. There are a number of attacks demonstrated against practical systems, and so there needs to be a very careful approach taken to analyze the security from all angles. Once a system is developed and is targeted towards commercial applications.
And what if it just breaks? What if it can the rest of the capsule just be digested, or what happens in that situation?
So for the capsule we developed, ours is not dissolvable, so it would just pass out normally. And in the case of the drug delivery star type devices, I could pass that over to Dr. Traverso.
So if the dosage form starts to break up, then its ability to reside in the stomach, is limited, and so it starts to pass through the body.
And that's perfectly safe, or we hope it will be.
That's right. And so there are certain portions of that dosage form that have safety linkers. And so if the dosage form is in the small intestine specifically, it also starts to break up even further in an accelerated fashion.
So say with your reservoirs, if something went wrong and all of them opened, this is a different device, but if all of them opened at once, is there some kind of system or some kind of protection against that happening? So
that's a great question. So there's a couple of things I think that you're getting at, and that is, the concept of dose dumping. And this is particularly at the regulatory level a concern and something that always has to be addressed with depot-like systems. So with respect to the STAR, the drug is essentially impregnated in the solid polymer. So there isn't really the ability to essentially like open a bottle and all of a sudden that to come out. In the other sort of concept, the work that we did together with Dr. Nadeau, there, we show the proof of concept of having wells essentially that are sealed with a gold film. And so again, you have a discrete holder of drug, and but part of the testing, if that were to move forward into for a clinical trial, would be to show that not all of them would open and create the toxicity, because absolutely you're right. I think that would be devastating. So that's part of the engineering challenge there whenever you have sort of those discrete wells.
Cool. Another interesting question: can you have two-way communication with the ingestible device once it's inside? Can you tell it when to release, or have some feedback there?
Certainly, you could envision having a receiver. Our device didn't have, but if you added a receiver to the capsule, you could envision being able to send a command from external to, as you mentioned, release the drug or perform a sensing task. I think this goes also back to the question about security. As soon as you have the reverse link, the requirements for security increase, and so part of the challenge there would be incorporating that and demonstrating that it is secure, and that's important work to do moving forward.
Right.
Karen, I see a question here about cost and whether mentioning that these are not cheap, and even if they go generic. And I think it's important to highlight the cost that's associated with non-adherence. And so probably one of the most dramatic examples of non-adherence that I've encountered is in the setting of a heart attack. So there was a very nice trial that came out of the New England Journal at the end of 2011, where a group asked the question, if, let's take all patients that have a heart attack in this particular institution and divide them into two groups. One, we give them the medications after the heart attack that we know will prevent complications and another heart attack, and give them for free to them, okay? And then the other group, the other half, were randomized to their standard sort of cost coverage with their insurances. So when you looked at both groups, the adherence rate difference, it was approximately 5%, and that 5% was somewhere around like 44 and 49% adherent at 12 months after myocardial infarction, which I think, or at least I would perceive as being a fairly significant life event. And so what we and I should say that 5% difference already had a significant effect with respect to complications and recurrent heart attacks. So the cost of non-adherence is really tremendous. And I mentioned at the beginning the magnitude of the problem in the U.S. alone. And so although I think there's probably a cost in the development of these new systems, there is also significant cost savings that are associated with ensuring and really making it easier for patients to take their medication. And I didn't go over this data, but there's data actually very specifically looking at what happens to adherence or compliance when a drug is dosed once a day, once a week, or once a month. And the adherence increases with every step. And so we know from multiple trials that when the medication is dosed a little bit more infrequently, that the likelihood of the subject, of the patient, of taking that medication does go up. So just speaking to that participant, it's I think they're right. There is an increased cost, but there's also significant potential for cost savings by amplifying the ability of a patient to take medication. And I think the other thing to consider is sort of different disease states. So there are certain conditions, for example, psychiatric illness, where sometimes the condition itself may impair the ability to remember to take the medication. And so, I think also there are certain populations that may be even more at risk. And that was, part of the interest from the Gates Foundation.
Do we have a sense of how much these cost, what we're talking about?
So, for the STAR systems, it's still at an early stage. The actual cost of goods is, on the order of the $1 range or multiple, single dollars. And it's not, but it's hard to tell, until we're closer to scale to have a good sense of where that will be. It may be in the tens of dollars. I'm honestly not sure. But it's, our sense is, at least from all of our conversations and work in this area, is that it'll be on par with other sort of depot-like systems that are more in the, that are out there and available. And so, that's our sense today. But I think the piece that often, I think isn't appreciated as well is the cost savings that are associated with maximizing adherence.
Right. And piggybacking on that, how long do you think it'll take till these are commercialized?
No, that's a great question. So we've co-founded a company. It's been around now for 2 years. And so, as far as having a product on the market, it's been on the order of years, not decades, to give you a sense.
Cool. And what about any interest from app developers for the personal information interface on these? Would I access this through my Tufts Healthcare account? Someone asks 5 years from now or whatever.
Phillip, would you?
Sure. Yeah. No, I could take that one. We've looked at building apps on phones to be able to interact with the capsules, and we have a recent demonstration of being able to receive temperature information from inside the stomach on a phone, and you can imagine that information being sent over the cloud to the patient's health provider. And to enable them to follow up and keep track. I think one of the most interesting things that's happening now with the cloud and big data, and also coupling that with the kinds of devices that we're developing now, is the ability to monitor and record for much extended periods of time, capturing data that is has not been captured before. You can imagine if the doctor is interested in looking at your ECG trace, they send you home with a recorder that you wear for perhaps a couple of days that records short segments whenever the system detects that there is an anomalous waveform. If you're able to record ECG information on the order of weeks and months and do it very unobtrusively, and put it into a machine learning algorithm along with data from a lot of different patients coupled with their outcomes, you can imagine that this could be a very powerful new tool that would enable new types of diagnosis to take place. So that's another interesting direction that's coming online with the medical information.
Great. Are there any clinical trials that are active right now or planned right now, or are we still at the lab stage here? For either of you.
So for the STAR, those are being planned, and so those will be happening soon. For the ingestible, the work that we did with Dr. Nadeau, that's still at the research stage at this moment.
Okay. And somebody asked, do you see any way to implant one of these devices so it can remain in the GI tract indefinitely? Is there a way to keep it there, keep it going?
No, so certainly, I think that's a great question. We are looking at other technologies that may be more amenable for implantation, and and certainly that could be done endoscopically, without essentially as a minimally invasive procedure. So absolutely, that's something that you might see even soon, sooner than yes.
Cool. What about using an RFID type device technology to pulse batteryless technology? They're saying, for example, an external powered receiver would not only receive but also send a pulse to trigger or power the sensor. And what are the applications for that compared to the lemon battery approach?
Yeah, that's also an interesting direction. The biggest challenge there is that being a gastric capsule, it's quite small, and there's not a lot of room for an antenna there, and the power is somewhat directional, and so when it's moving around in the GI tract, it's more difficult to maintain a link. Some of the work that Dr. Traverso's group has done on the midfield wireless power is interesting in the sense that it lets you transmit the power deeper inside the body. So it would let you reach an ingestible device. We still need to do more work on the directionality of it, keeping track of where it is in the GI tract and beaming the power in that particular direction. But that is one of the ways that we could transmit power from external. I think the advantage is that you'd have a potentially limitless source of power, as long as you recharge the external device or plug it in periodically to charge it. Whereas systems like the lemon battery, of course, run out once the materials inside have expired. And so that's an interesting direction. The biggest challenge is maintaining the link, so to speak, the directionality of the power despite the movement of the capsule.
Great. And this is our last question because we're running out of time here. But what are the biggest challenges that you see lying ahead? Are they chemical? Are they mechanical? What are the big issues going forward?
So I'm happy to take at least part of that. So with respect to gastric residence, that's really a mechanical challenge, and that systems that reside in the gastric cavity need to have the mechanical integrity to withstand the compressive forces of the stomach and then also the food stream, and then ensuring that with respect to, for example, drug delivery, maintaining stability of the drugs can sometimes be a challenge, and that's very drug specific. So so chemical specific. And on the electronic side, I'll let Dr. Nadeau.
I think sure, yeah. Like for the packaging is probably the biggest challenge. Getting all of the electronics designed and integrated is one thing, but another is making it compatible with the stomach environment and making it last for long term. Also preventing leakage of materials inside. It's a very significant challenge, and one that still for at least for the capsule that we've demonstrated would benefit from improvement.
Well, sounds like you're doing that all the time. And it's really interesting work. Thank you so much for sharing it with us today. And on behalf of the Alumni Association, thanks for tuning in to the Faculty Forum Online. And any questions we didn't get to, we'll forward to the panel. Also, please tweet about today's chat using the hashtag #MITBetterWorld, and send any follow-up questions or feedback to alumnilearn@mit.edu. Thanks for watching.
Thanks so much.
Thanks so much.
Bye.
Bye.